My name is Luyi. I'm a graduate student studying biology, and today I wanted to talk to you about what we can learn from biology about this fascinating molecule called RNA, and how we can use this knowledge to build some amazing tools for biotechnology. I want to start off by talking about water. We all need water to survive. It keeps us alive. But it's not just having the water that's important. We need it to be clean also. So we need to put a lot of thought into what's in our water. Take, for example, something we know is added to our drinking water. In the United States, our water has been fluoridated since the 1960s to prevent teeth decay and improve dental health. But something we might not think about every day is that having too much fluoride can also be harmful for us. In other regions of the world, for example, like Cartago in Costa Rica, too much fluoride in the drinking water can lead to serious health concerns like dental and skeletal fluorosis, which causes your teeth and your bones to break down. This region has been tested and documented for having too much fluoride in the water. But the thing is, there are even more at-risk regions of the world that just don't have the necessary resources to even know if the water is safe to drink or not. It requires time, expertise, expensive equipment, which isn't always that accessible. So that means we need a cheap, fast, and easy way to test for safe drinking water. This is a picture of a member of my lab, Matthew Vereslov. He's in Cartago in Costa Rica collecting a sample of water. He's testing these tools that we designed in our lab by Walter Thavaraja and Adam Silverman to do exactly this. He's using these test tubes that contain these reactions, where all you need to do is add a single drop of your water, which will produce a bright yellow color change if there's fluoride in the water. These reactions were made in our lab, where we mix the reactions, freeze-dry them into a powder form onto paper discs or smaller tubes, which then just require that drop of water to rehydrate so the reaction can run. We call these tools biosensors, sensors made using biology. And how does this biology work? This biology works using a molecule that we call RNA. A lot of us are more familiar with thinking about DNA. We know this as a molecule that's responsible for carrying the genetic information that makes each of us and every single living organism a unique individual. When it comes to RNA, we can think of it as kind of like DNA's cousin. It's similar in some ways, like what it's made of and what it can do, but it's also different in some very significant ways. For example, one of the biggest differences is that while DNA is made up of 2 strands, RNA is made up of only 1 strand. Because it's made up of only 1 strand, it means it has the room to be flexible enough to fold into some really interesting and complex structures. And these structures are what fascinates me. I'm trying to understand how RNA folds into these structures, because it's also these structures that make our biosensors work. Our biosensors, they use these RNA molecules that fold into a structure called a ribose switch. Ribose switches fold into very specific structures that fit like an exact puzzle piece match to very specific molecules. In our case, our ribose switch is the exact match for a fluoride molecule. So that means if there's fluoride floating around the structure, it'll fit and match the ribose switch, which will then cause it to switch on a protein that will produce a bright yellow color. And this lets you know by eye that there's fluoride in the water. Ribose switches are found all throughout nature, a lot of it in bacteria, but like a lot of RNA structures that we know of, it's still kind of a little mysterious how exactly it works. It's kind of mysterious just like how when you pull out earphones from your pocket, it's suddenly a tangled mess and you have no idea how it got like that in the first place. And then you have to start to untangle it. And that's what I'm trying to do. I'm trying to untangle these RNA structures to understand how they folded into these shapes in the first place. And here's how I'm trying to understand them. RNA is made up of sequences that consist of 4 letters that kind of act as building blocks. These letters, they sometimes like to be by themselves, and other times they like to pair up with others. And they have preferences also. U's like to be with A's, and C's like to be with G's. And these preferences can also change depending on what's neighboring the letters also. And so when you take into account all the preferences of every single individual letter, and you look at a complex structure like our fluoride ribose switch, it's not just a random tangle anymore. There's actually a pattern behind how these letters like to rearrange itself into a structure that has a purpose. And this is the pattern that I want to try to understand. how does the RNA sequence lead to its structure, and how does this structure lead to its function? Because if we can understand this pattern and the connections between them, that means we can take a ribose switch and intentionally change the sequence to change the structure and change how it works. We can engineer the ribose switch to respond to different molecules or to turn on different proteins that will produce different colors. And this opens up a whole wide range of biotechnology tools that we can develop that can be used way beyond just our labs. Biology is complicated and complex, but I think it has so much to teach us. If we can learn all that we can from biology, especially about RNA, then the possibilities for developing accessible biotechnology tools, like for accessible water quality testing, makes it all the more worthwhile for us to try to understand.